Welcome to the FOCIS podcast, brought to you by the FOCIS Communications Committee.
Well, hello, Dr. Vivier. Thank you so much for being here today. To start off this interview, what research have you conducted that you're most proud of, or any papers that you've been working on recently?
Yeah, we are working on natural killer cells, NK cells. So I think that those kind of cells require some introduction. So they are lymphocytes of the innate immunity. And so we have been studying those cells since quite a long time already. And I think what is interesting is to try to translate the result of basic research on natural killer cells to know what they are doing, what they are coming from, in particular in the context of cancer. So what we've learned is that NK cells can recognize tumor cells because they express activating receptors that recognize ligands, which are not expressed on normal cells, but preferentially expressed on stress cells, including tumor cells. So based on that, and we have, we have participating to these discoveries, but this is a collective venture, so to speak. We have been able to try to translate these findings into new assets for a better treatment of patients with different kind of cancer. So we are developing two kinds of drugs, and this is in particular in collaboration with Innate Pharma, a biotech company located in France. And so one category of assets are immune checkpoint inhibitors, like the anti-PD-1, anti-PD-L1. But what we are targeting here is actually is NKG2A, an inhibitory receptor, which is expressed both on NK cells and T cells. And the idea is when you block the interaction between NKG2A and its ligand, then you will unleash the cells, allowing them to be more efficient to control tumor burden. And so we are now, when I say we, it's Innate Pharma, in a partnership with AstraZeneca in a phase 3 pivotal clinical trial in non-small cell lung carcinoma. On the other side, we are also trying to activate NK cells through engagement of these activating receptors that I briefly mentioned. In particular, we are developing a new therapeutic class that we are calling ANKET for antibody-based NK cell engager therapeutics. And those molecules will engage NK cells through activating receptors, namely NKp46 and CD16, and then targeting a tumor antigen expressed on tumor cells. So we have we are in the process of testing in phase 1-2 clinical trials 3 of these molecules, one targeting CD123 for acute myeloid leukemia, another one is BCMA for multiple myeloma, and finally, there is a proprietary Innate Pharma molecule which carries also non-alpha IL-2, interleukin-2, which is able to induce NK cell proliferation, and which is tested in non-Hodgkin B-cell lymphoma.
Right, well, that sounds very interesting. So then, do you want to discuss any presentations or posters that you presented this week at FOCIS 2025?
So at FOCIS 2025, I was presenting new stories that we have in the lab, and also some of the update of what we're doing with Innate Pharma. So for the lab part, the academic lab at the Center for Immunology in Marseille-Luminy in France, we've been able to put together a consortium of 20 labs across the world to try to document and understand the diversity of NK cells. And so this consortium is called Meta-NK. The paper was published in Nature Immunology as of last year. And we've been able, using single-cell RNA-seq and site-seq, to perform a meta-analysis of the diversity of NK cells and came up with a very easy-to-remember nomenclature of NK cells, namely NK1, NK2, and NK3. But there is more than that, because we could also anticipate or propose what could be the function of these different subset of NK cells, and in healthy conditions, but also in tumor conditions. So based on that, I've been presented at FOCIS 2025 a new story that we have with Alfred Zipelius and Clara Serge in Basel, in which we concentrated on non-small cell lung carcinoma. And we could identify NK1, NK2, and NK3 as being present in the tumor of the patients using samples from cancer patients. But more than that, we could identify specific subset of NK cells which are tumor-associated and not present in the healthy lung. So we call these cells TANK for tumor-associated NK cells, and they come basically in 3 flavors. And we've been able, using single-cell technology again, single-cell RNA-seq and ATAC-seq, to document what these cells are about and what are the positive function. And we could see that there is a subset of NK cells expressing CD39, which can be really unleashed by anti-NKG2A antibody, such as monalizumab. So I think it's very important, I think, because it tells us that in non-small cell lung carcinoma, there's a subset of tumor-associated NK cells that can be unleashed with proper reagents, such as monalizumab, hopefully leading to a better control of the tumor.
All right. Well, thank you so much.